last time we briefly discussed the diversity of organisms in general. It was mostly focused on the different ways we classify organisms based on their relationships and determine how close those relationships are. We talked a lot about taxonomy, a lot about phylogeny. We didn't do much discussion of any actual specific organisms, though. So today, we're going to begin with chapter 13. That focuses on the diversity of microbes, fungi, and protists. So microbes, or microbial life, those are microscopic organisms. That includes an enormous amount of diversity because that includes the bacteria, that includes the archaea, that includes many eukaryotes, such as a lot of fungi and a lot of protists. So there's a lot to do in microbes. First off, prokaryotes. I briefly talked about these earlier in the semester. So everyone should hopefully remember that prokaryotes are microscopic, they're mostly unicellular. There are a few cases in which they can be colonial, but as far as I'm aware, there are no truly multicellular prokaryotes. They're also extremely simple. That's one of the things that sets these apart from eukaryotes, from organisms like us. They don't have a lot of the features that are found in more complex life. So they lack the true nucleus. That's the main designation. That's what prokaryote translates into roughly. It means before nucleus. These were the earliest life forms to exist on Earth. We are a descendant from prokaryotes. Luca, that last universal common ancestor, was a prokaryote. And this includes two separate domains. I've also mentioned that back when I was in college, it was generally considered to be one domain, but recent genetic evidence has given us substantial cause to separate the bacteria from the archaea. We now consider those to be two separate domains because we've determined that they are different enough that it's not appropriate to group them together. Now, what the prokaryotes does not include is fungi or protists, which are two other subjects of this chapter. Both of those are eukaryotic organisms. So fungi and protists, even the unicellular ones, are still far more complex than any prokaryote. You'll still see a true nucleus, you'll see membrane-enclosed organelles. They're much larger and much more complex, and much more closely related to us than they are to prokaryotes. Now, when it comes to the niche in general of prokaryotes, it's just about everything. These things are just about everywhere. Almost every surface on Earth is home to prokaryotic organisms, including you. You have prokaryotes living all over you and inside you as well. Although in biological terms, most of them aren't considered to be inside you, because in biology, nothing is really considered to have entered your body until it crosses a membrane. If I were to swallow a quarter, I would eventually pass it out the other end. And biologically speaking, that quarter will never have been inside of me. It just passes through a tube, runs through the middle of it. But in pejorative terms, people would say that quarter is in. So in terms of most of the prokaryotes living, quote unquote, inside your body, they are somewhere in your gut, in your digestive tract. But if they never pass through a membrane, a lot of people who are really pedantic and sticklers about semantics would say that they're not technically inside. Interestingly, there are far more prokaryotic cells living either on or surrounded by your body in your gut than you have actual human cells in your entire body. Way more. Part of the reason this is possible is remember that prokaryotic cells are much, much smaller. Eukaryotic cells can be 100 times or 1,000 times larger than a prokaryotic cell. So the general volume of your body is primarily your own cells. But the cells themselves individually are outnumbered by the prokaryotes that you are hosting. If you're a germaphobe, that may keep you up. They're also found in extremely harsh environments. Bacteria and archaea, certain species can live really almost anywhere. They're found in hot springs. They're found in hydrothermal vents on the seafloor. They're found in really salty and really acidic environments where basically nothing else can live. They're also extremely important in nutrient cycling. So bacteria especially are an important link in the biogeochemical cycles that recycle things like carbon and things like nitrogen that allow chemical molecules to be returned to the base of the food chain for reuse. Without these bacteria helping with nutrient cycling, we wouldn't be able to fully decompose dead organic matter back into inorganic matter for producers to use. Everything would pile up and the system would grind to a halt. So when we talk about early life on Earth, when I say that prokaryotes were the first things to exist, Earth looks very different in its early days. It's estimated to be roughly 4.5 billion years old. People much smarter than me have arrived at that estimate based on ages of meteorites. So none of the rocks on the surface of Earth are as old as Earth. It may not seem like it, but all those rocks continually go through cycling. They get recycled in the hot core of the Earth and pushed back up to the surface as new, younger rocks. Some of the meteorites that have struck Earth's surface, though, are, actually, is it meteorites or meteors that make it to the surface? I can never remember. Not important. Stuff from outer space that hit Earth. Those things are often not recycled the same way that Earth rocks are. So by dating those and seeing how old the oldest extraterrestrial objects we can find are, we can roughly guess how old the Earth probably is. Now, the Earth being 4.5 billion years old, we have evidence to suggest that the origins of life on this planet probably happened about 4.2 to 4.3 billion years ago. So the Earth was probably only a few hundred million years old when the first living organisms came to be on this planet. You may or may not be aware that the early Earth had no oxygen in the atmosphere. We had much higher proportions of carbon dioxide than you see today. It was also much hotter. That's because carbon dioxide is much more effective at trapping heat than oxygen is as a gas. That's why it's considered a greenhouse gas. More chemical bonds equals higher potential for heat. So the early Earth's atmosphere was quite a bit warmer and contained no oxygen. Therefore, only anaerobic organisms could have survived. Anaerobic are things that do not need oxygen. So the first life on Earth survived in complete absence of oxygen. Early phototrophs, those being things that are capable of photosynthesizing to make food, those probably appeared about 1 billion years later. Pretty long time. So there was no oxygen in Earth's atmosphere for roughly the first billion, maybe 1.25 billion years that the planet existed. These early phototrophs ended up evolving into cyanobacteria. These are prokaryotic organisms. They're in the kingdom bacteria, and they are capable of carrying out oxygenic photosynthesis. So when something photosynthesizes, you guys probably, hopefully all remember that it needs some source of electrons to keep the process moving, right? There are several different sources of electrons that can be used, but the most common one in organisms currently existing is water. So whenever something uses water as an electron source for photosynthesis, it ends up producing O2 gas as a byproduct. So as these cyanobacteria evolved and started using more and more water for photosynthesis, they started oxygenating the atmosphere, gradually pumping more and more O2 into the air, which eventually allowed for the evolution of anaerobic organisms. Aerobic organisms, things that use oxygen for cellular respiration. It also had some other pretty important effects on the atmosphere, which we'll get into in a moment. So most of these early organisms are thought to have lived either very deep in the ocean or deep below the Earth's surface. Can anyone guess why? Why do we think that was the case?
Surface was still unlivable.
Why was it unlivable?
No atmosphere.
Not that there was no atmosphere, but the atmosphere had no oxygen. And what's oxygen good at doing in the atmosphere? I'll be impressed. Well, I won't be that impressed. You just need to read the chapter like I asked you to. But oxygen in the atmosphere is really good at filtering out UV radiation. You guys have all heard of the ozone layer, right? Ozone is a relatively unstable compound that it's a molecule made of 3 oxygen atoms. So when O2 is excited by UV radiation, it often dissociates. It absorbs the radiation, the bond starts to wiggle, and then it breaks. So you have all these free oxygen atoms, which will, in many cases, spontaneously bond with each other to form ozone, O3. That is going to absorb even more UV radiation and break again, may go back to O3, may go down to O2 with a free oxygen atom. But either way, having large concentrations of oxygen in the atmosphere is what filters out UV radiation. That's what keeps the sun from frying everything alive. Just remember that UV radiation has a short enough wavelength and therefore high enough energy to penetrate into our cells and damage our DNA. It's also thought that these early life forms were adapted to very high temperatures. And that is because during the early 1 billion or 2 years of Earth, volcanic activity was extremely strong. The planet was much, much hotter than it is today. There were a lot more eruptions and a lot more shifting of plates of the surface. So anything that would have lived back then would have to be pretty tolerant of very high heat. Now we think that early life probably existed as microbial mats. A microbial mat is just a very large biofilm, and a biofilm is just a bunch of different prokaryotes living on some surface together. Different species that live together, and they're held together by secretions that are mostly polysaccharides, but will also contain some proteins. So in nature, a lot of the time, prokaryotes tend to grow as these microbial mats. We still see that. We still see biofilms everywhere. Now, describing these organisms that exist in ocean depths or exist under extremely high temperature, these days, we tend to call these things extremophiles. So extremophiles are any organism that inhabits a very extreme environment. And that may be extreme heat, that may be extreme cold, or salinity, or acidity, or alkalinity. For example, the organisms, yes?
Can darkness be one?
I don't know if you would consider darkness an extreme environment, but I mean, someone else may give you a different answer to that question. Yes?
What's that hydrothermal vents?
So the hydrothermal vents, good question, I was just about to get into that. So hydrothermal vents are basically little cracks in the surface of the earth around the bottom of the ocean. And they leak extremely hot water that's been heated by the Earth's core. When I say extremely hot, I mean in some cases it gets up to about 200 degrees Celsius, which is double the boiling temperature of water at the surface of the Earth. The only reason the water stays liquid is because of the intense pressure that it's under as a result of being many, many kilometers below the surface. So the water above it is pushing hard enough on it that it actually keeps it from boiling into a vapor. It's really interesting. But there are organisms that live all around these hydrothermal vents. So those things have to be able to withstand exceedingly high temperatures. Prokaryotes, obviously, are very different from eukaryotes. I'm sure I don't need to remind you guys of that. But they do have things in common, right? Yes, they do. So remember that all living cells have 4 things in common. Anybody remember what those are? Yeah, membrane. No life can exist without some sort of membrane separating it from the external environment. All cells also contain cytoplasm. That's the gooey fluid inside the membrane in which all the internal structures are suspended. Yeah, they all also have genetic material, meaning DNA and RNA. And the last thing all cells share is ribosomes, yes. That's what separates viruses from things that are technically alive. So remember, ribosomes are the organelles that make proteins. If you can't make your own proteins, you can't reproduce by yourself, and therefore you're not alive. So anything we do consider to be alive must have those. Now, most of the prokaryotes can be rather broadly categorized by shape. There's a whole bunch of different ways to break these up. Some of them are more useful than others depending on what type of questions you're trying to answer. But very commonly, we'll classify prokaryotes by their shape, and there are three main categories that most of them generally fit. And they look like that. The first of which being the cocci. Those are spherical bacteria, or archaea. We also have the bacilli, which are rod-shaped in the middle there. And then finally, the spirilla, also called spirillum. Those are spiral-shaped. They kind of look like a corkscrew. So it turns out that a pretty good number of prokaryotes can be placed into one of these three categories. That's particularly useful for classifying species, because that's going to be one of the easiest things to observe about these if you have a good enough microscope. Now, beyond that, obviously remember that prokaryotes are much simpler than eukaryotes. They don't have any membrane-enclosed organelles. Even their ribosomes, even though they have them, are pretty different from eukaryotic ribosomes. They are much smaller and much simpler. And you'll never see a nucleus or a mitochondrion or, I don't know, Golgi apparatus inside of prokaryotes. They also have a single circular chromosome. So that's in contrast to, for example, our 23 pairs of linear chromosomes. They have a much simpler genetic code, a lot less genes than they need to pass on. And most of them have cell wall. You see a lot of eukaryotic organisms that also have cell walls, generally in the kingdom of fungi or plantae, but they are quite different from the cell walls found in prokaryotes. Even within the prokaryotes, if you split it up between the two domains of bacteria and archaea, the cell walls vary quite a bit in their structure. So the cell wall generally serves the same purposes that it does in any other organism with a cell wall, even though it's put together quite differently. It basically forms a protective layer that surrounds the outside of the cell. So remember that cell walls always exist outside of the cell membrane. But also remember that anything that has a cell wall still has a cell membrane. The wall can't take the place of the membrane. So one of the things that it does is it gives that cell shape and rigidity. That's what allows plants to stand upright even though they have no bones. As I already said, located outside the cell membrane. And it varies greatly in composition between bacteria and archaea. But beyond that, it also varies a lot among different species of bacteria, so even within that same domain. The two main types of bacteria classified by cell walls are the gram-positive and gram-negative. So in addition to the overall shape of the bacterial cell, this is another way that we can classify bacteria. It's so named by the process that determines whether it's gram-positive or gram-negative, and it's a staining process. It involves a specific series of chemicals applied in a specific order that will result in either the cell being stained purple or being stained pink. If it's stained purple, that's a gram-positive cell. If it's stained pink, that's a gram-negative cell. Basically, what that depends on is the composition of the cell wall. So gram-positive bacteria have a very thick peptidoglycan wall that exists outside of the cell membrane. This here on the left is an example of a gram-positive bacterium. And these blue bricky-looking things are layers of a biological material called peptidoglycan. You know, I charge the battery for this remote. I need it. So on the right here, we have an example of what a gram-negative bacteria can look like, where it has a very thin peptidoglycan cell wall, and outside of that is another layer of membrane. Now this is useful for a number of reasons. Particularly, it's useful in, it's clinically relevant. So if you go to the doctor with some kind of bacterial infection, one of the first things they'll do if they're trying to figure out how to treat it is they'll perform a gram stain test on a sample of the bacteria. That tells you what type of cell wall it has, and that can inform decisions based on what kind of antibiotic the doctor wants to prescribe. Because a lot of antibiotics generally are designed to attack specific mechanisms within the cell wall. So some kind of antibiotic that is really good at disrupting the construction of peptidoglycan is going to be really good at killing gram-positive bacteria, for instance. So it's not just for academic purposes that we want to classify these things. It can also be really clinically relevant in cases of infection. Yes?
Kind of nasty, but I had strep throat one time and they took like a swab basically of your mouth. Is that the type of test that they would do to it to determine
So
whether or not?
there's a bunch of different things they can do with that. If you get a throat swab to test for strep, the first thing they're going to do is apply it to a plate, an agar plate, and allow the bacterial colony to grow for a while. So streptococcus or strep throat is caused by a bacterium called Streptococcus aureus, which is pretty easily recognizable just from looking at the colonies. Because anybody in here speak any Latin or French? Okay, so it gets the species name aureus from the Latin root for golden, because the colonies of Streptococcus aureus have a really nice golden color to it. So that one can be fairly easily diagnosed just visually. But another thing they might do, if it turned out that maybe it wasn't strep, is do a gram stain. So then they can have a better idea of what they're dealing with. Any other questions at this point? Anybody in here ever had strep throat? I have a bunch of times. I'm sure most of you probably have. Really not fun at all. All right, so I mentioned that the cell walls also vary in composition between bacteria and archaea. So the archaeal cell walls don't contain peptidoglycan at all. Instead, you can have 4 different types of cell walls within the archaea that are composed primarily of either something called pseudopeptidoglycan, which is similar but not the same molecule. They can also be composed of polysaccharides. You guys remember what polysaccharides are? Things like starch, cellulose, long chains of sugars. They can also be composed of glycoproteins, which are protein molecules that have associated sugars built into them, or other carbohydrates. And there's also something called S layers, which are surface layer proteins, which is where the name comes from, that certain archaeal cell walls are also composed of. Before we move on, anyone have any questions about cell walls in general, bacterial or archaeal cell walls, gram-positive versus gram-negative?
So archaeal cells can never be gram-positive or gram-negative?
I actually don't know if they respond to a gram stain or not. I don't think it would generally be a relevant test.
Okay.
But that's a very good question. Now I'm kind of curious. I'll have to ask somebody. All right, so reproduction in prokaryotes. We already talked about this at the end of the lecture about mitosis. You guys remember how this all works? So you see primarily asexual reproduction in prokaryotic organisms. These things are not complex enough for the most part to reproduce sexually. They don't produce gametes, they just divide. So it's a cellular division that takes place through a process that is called binary fission. Looks like that. You guys have already seen this. Oh, laser pointer's not working. Anyway, it's very similar to mitosis in principle, but it's a lot simpler. Largely because there's only that one single circular chromosome that has to be copied. So it's not nearly as complex as having to copy, 10 or 15 or 20 or in our case 23 pairs of chromosomes, which then have to be neatly arranged and separated and make sure that the right number of copies end up in your cell. In this case, that little ring of DNA just replicates itself and one goes to either side, and then the cell pinches off in the middle. Now, one of the similarities it shares with mitosis is this doesn't really provide any opportunity for genetic recombination. We talked about that during the meiosis lectures, talking about sexual reproduction and how one of the big advantages of that is it allows for great increases in genetic variability through things like crossing over and things like independent assortment and random fertilization. So sexual reproduction is really good at increasing genetic variation in a population. If you divide through binary fission, you don't get those opportunities. But prokaryotic organisms can increase their genetic variation through several different mechanisms. One of which being something called transformation. Basically, all transformation is in this context is a prokaryotic cell will take up some DNA that is found somewhere in its external environment. This could have been from a destroyed cell, this could have been DNA that was just excreted out of a cell, but basically it just finds some DNA and it's like, oh, this looks interesting, I'm going to take that, and it incorporates it into its own genome. If you're a simple enough organism, you can do things like this. Another way is transduction. So we talked about how viruses reproduce briefly. Anybody remember how they do that?
They inject their DNA into the cell.
Yeah, so viruses will infect some specific kind of cell. They will inject their DNA, they will hijack that cell's ribosomes to make new viruses. When they do that, they often end up transferring little bits of DNA from one prokaryotic cell to another. That DNA that gets transferred can also be absorbed into that really simple genome that consists of one singular chromosome. There's also something called conjugation. This is when DNA can be transferred directly between two prokaryotic organisms by direct contact. So these are three different ways where prokaryotic organisms can come up with some recombinant DNA.
Would these be examples of maybe how eukaryotic cells ended up with the mitochondria? Because
We're going to talk about that later, but probably not. Although there also is no completely certain answer to that question because the prevailing current theory, endosymbiosis, is still really poorly understood and we don't really know how all it works. Anyway, so this may seem like a really clumsy way to increase genetic variation, but cycles of binary fission can be exceedingly rapid. In some species of prokaryote, the generation time is as short as a few minutes. So over the course of just one day, you could have hundreds of generations. If you couple that exceedingly short generation time with these three mechanisms of somewhat clumsy genetic recombination, you can see how prokaryotes, even without sexual reproduction, can still evolve very rapidly. So we're great at creating recombinant chromosomes, recombinant DNA. We're great at giving rise to new genetic variation. But it takes us a really long time to reproduce. I mean, the fastest humans could possibly reproduce would probably be 12 or 13 years as far as generation time. And that's weird and gross and we don't do it. Generally, it takes us more like 20, 25, 30 years before anyone starts reproducing. Although I did have a friend in college who had her daughter at 15. So that does happen. But for the most part, human generation times are quite a bit longer. In fact, even if you are reproducing at the age of 12, that's still enough time for hundreds of thousands or maybe millions of generations in some of these prokaryotes. Remember that the big key for a population evolving is more and more generations, because individuals can't change their genome. Any questions about that? Yes?
I imagine this could also work against you, though, right? With how fast it is reproducing.
So it's pretty rare that natural selection can actually be detrimental to a population, unless there's some extreme and very drastic and fast change in environmental conditions, because natural selection is generally only going to make you better adapted to the environment you live in. So it's going to amplify traits that are beneficial, and those traits may not always be beneficial, but it's pretty rare that they will rapidly become detrimental. Does that make sense?
Yeah.
So increased rate of evolution is very rarely going to be harmful for a population. Oops, wrong button. Anything else before we move on? So that covers prokaryotic reproduction. Prokaryotic metabolism. You guys remember what metabolism is?
Energy.
Yeah, energy processing. An organism's metabolism is just the sum total of its energy processing reactions. Prokaryotes, being as diverse as they are and occupying all these extreme and diverse niches, also have extremely diverse methods of obtaining energy. So one of the big ones is decomposing dead organisms. That ties back into their importance to nutrient cycling. So when you die, you're going to get eaten, unless someone burns you, which we do now, but when you die, if you just died naturally and rotted naturally, you would be eaten by small animals, you'd be eaten by worms, insects, then they would be excreting smaller broken down parts of you, but it would still be organic matter. The things that actually turn organic waste into inorganic nutrients are things like prokaryotes, generally bacteria. They will, break down ammonia into constituent nitrogen or hydrogen. Another way they can get their energy is by growing inside other organisms. Like right now, there's a whole raft of bacteria in my large intestine, and they're getting the donut that I had for breakfast. That is now their food. Some also use light. Phototrophy, that is seen as prokaryotic organisms, primarily the cyanobacteria. And there's some other really cool stuff. Some of them are able to use inorganic compounds, like the methanogens. Some of these extremophiles live way down deep by the hydrothermal vents. These things use inorganic chemical molecules to produce organic chemical energy. It's really cool. But the big take home here is that prokaryotic metabolism is exceedingly diverse and varied. They occupy all these different niches which necessitate the evolution of different mechanisms to obtain energy. And they've been very good at making that happen. Any questions on this? Okay. So, when people think of bacteria, a lot of the time what they think of is something gross. It's going to make you sick. However, not all prokaryotes are pathogenic. Pathogenic means disease-causing. So not all of them do this. In fact, no archaea are known to be pathogenic in humans. All of the pathogenic prokaryotes that infect humans are bacteria. Although even among the domain bacteria, most bacteria are either mostly or completely harmless to us, as long as they're where they're supposed to be. Like those bacteria that are mowing down on that donut right now. As long as they stay in my large intestine, everything's great. However, if I were to suffer some sort of awful injury to my abdominal cavity and puncture the lining of my large intestine and those bacteria were to spill out into the rest of my body, that would be extremely harmful and would probably kill me unless I got to the hospital right away. But as long as they stay where they're supposed to be, everything's great. Now, among the pathogenic bacteria, they're known to cause a pretty wide variety of diseases in humans. A great example is tuberculosis. The bacterium responsible for causing tuberculosis is extremely virulent. It's very good at causing infections. In most cases, you would need hundreds or thousands of these bacteria inside your body before a full-blown infection could be established. Tuberculosis is virulent enough that one single bacterium is enough to cause a full-blown tuberculosis infection. The bubonic plague, that's another really iconic disease caused by bacteria. We think that was responsible for the Black Death in Europe in the 14th century that killed like a third of the world's population. Cholera, another one. Anybody ever play the Oregon Trail computer game? Were you guys too young for that? And you've died of dysentery, you've died of cholera, etc. This was a disease that was much more common hundreds of years ago, before we had really good sanitation techniques. Chlamydia, syphilis, these are also quite common. Syphilis can actually kill you. Be careful out there. Those are STDs. Many bacterial diseases, however, can be treated with antibiotics. So these are chemicals, like I mentioned earlier, that specifically target some mechanism inside the bacterial cell That does not exist in your own cells. That's why you can apply them wholesale to your entire body without hurting yourself and only harming the bacteria. So if you apply something, you ingest something that interrupts the production of peptidoglycan, that's going to harm bacteria because they need that to build their cell wall. But it's not going to harm you because none of your cells are producing peptidoglycan to form the necessary structure for survival. Antibiotics are really cool. But we've also really overused them. I mentioned a little bit about that when I was talking about the development of pesticide resistance in insects in the evolution lecture. It also works the same way with antibiotic resistance in bacteria. Now, without me telling you, do you guys understand why antibiotics can only be used to treat bacterial infections and not viral infections? Why can't we treat AIDS with antibiotics? Anybody?
Virus is a different structure.
Yeah, viruses are entirely different organisms. In fact, the bacteria are probably more closely related to us than they are to viruses. So you can't use antibiotics that are designed to target prokaryotes on some entirely different organism. That's why antibiotics cannot fight viral infections. Now, when it comes to antibiotic resistance, this is the result of decades and decades of us really recklessly using these wonderful antibiotics that we developed in the last century. So overuse. For too long, anytime someone had, any little illness, they would go into the doctor, and the doctor's like, all right, here's some penicillin, you're great, you're fine. But the more you use this, the more you apply these antibiotics, the more resistant the populations become. We also developed this really dumb practice of just pumping all of our livestock full of antibiotics to prevent disease transmission in our ridiculously, morally irreprehensible factory farms. I mean, we didn't even wait until the cows were sick. We just shot them up with antibiotics as a preemptive measure. So when that happens, just like with the pesticide application to the insect population, the ones that are the most vulnerable are going to be the ones that get killed. What that leads to is the resistant bacteria surviving and being able to reproduce with basically zero competition. What that leads to is that genetically conferred resistance being passed on to future generations and becoming more and more common in the population until basically everything has it. Staphylococcus aureus, which we were just talking about. This is a great example of a common bacterium that is found both on and in the human body. Wait.
No, this isn't strep, it's staph.
Yeah, it's a strep staph. I totally misspoke when I was talking about that one. Strep is not caused by this bacteria. Anyway, staph infection, you guys may have heard of it. Even if you haven't heard of this, you've probably heard of what we have recently generated through our overuse of antibiotics. So it used to be that staph infections were very easily treatable with antibiotics. If you had a small cut that got some staph in there, you would just get a shot of, I think methicillin was the most common one they used for it. Different antibiotics for different bacteria. But our overuse of that methicillin has given rise to something called methicillin-resistant Staphylococcus aureus, which is commonly called MRSA. You guys may have heard of MRSA. This is becoming more and more common, particularly in hospitals. Patients can get infected with this. It's also, I had one of my buddies actually get a MRSA infection. He's a personal trainer, and he got it from the gym equipment. This stuff's everywhere.
I had a roommate that had it, and they had to get his arm cut open, and they removed a portion of it.
Yep. Yeah, in extreme cases, if the infection gets out of hand, they sometimes have to amputate large parts of the body. And even in that case, it doesn't always cure it. People still die from this. So antibiotic resistance is becoming really serious. And it's sort of interesting because we enjoyed this short period where bacterial infections weren't really all that threatening. For most of human history, a bacterial infection was very deadly, but we basically stopped all that with the invention of antibiotics. Now that we've overused our antibiotics so much, it sort of looks like we may be gradually returning to a world where bacterial infections are very deadly and often fatal. Why am I pointing at this over here? So, before I move on, actually, any questions about pathogenic prokaryotes or antibiotic resistance? If you ever get prescribed antibiotics, make sure you finish the entire course. In addition to overuse, misuse of antibiotics is also largely responsible for giving rise to antibiotic-resistant strains. So basically what that means is you may have some bacteria in your body that are sort of resistant, like they have some resistance to that antibiotic, but if you apply enough of it, it will still kill them. So when you're given a course of something for some infection, and the doctor says, take these 3 times a day for a week, a lot of people, after 3 days, they start to feel better because it knocked out most of the infection. They're like, oh, I really need to finish this, so they throw it out. That allows more of the partially resistant bacteria to survive and reproduce and confer more resistance to future generations. So if you're ever prescribed antibiotics, make sure you finish the whole course on schedule. What's that? Oh, yeah?
Yeah, I ate E. coli one time, and it came back because my mom lost a bottle of
Oh, no.
antibiotics.
Yeah.
That sucked.
Yeah, E. coli actually used to be pretty harmless. Like back in the 50s, E. coli would at worst give you a case of traveler's diarrhea. But because we had pumped so many antibiotics into our livestock, we developed these really antibiotic-resistant strains that are now potentially fatal. So, oh yeah, I mean, remember last lecture when I was talking about binomial nomenclature? You guys have probably been using it your entire life, whether or not you didn't. E. coli is a great example. We'll get into that in a couple of slides. How much time do we have? Okay, so prokaryotes, as I said, are basically everywhere. They inhabit almost any surface on the planet, and food is no exception. Everything you eat has bacteria living on it. It also has other microbes, like fungi. Has anyone ever had some food start molding in the fridge? I'd be shocked if any of you hadn't had that happen. It happens all the time. Where did that mold come from? It was already there. By the time you even brought the food into your house, it probably had mold spores on it. It just took a fair amount of time for those spores to germinate and the fungi to reproduce. It works the same with bacteria. Any food you bring into your house is already going to have bacteria living on it. If you leave it in the fridge for a long enough time, those bacteria are going to have time to reproduce and multiply.
That's why we use the fridge, right?
That's exactly why we use the fridge. It slows down the generation time of those bacteria. It doesn't stop it. Even freezing it doesn't always completely stop it. But the lower the temperature is, generally the slower the reproduction is going to be. That's why things go bad faster on the kitchen counter than they do in the fridge. But that's why they still go bad eventually in the fridge. Food poisoning. Who's had food poisoning? I have, several times. It's awful. That's the result of eating something that is contaminated with some pathogenic bacteria. Remember that not all bacteria are pathogenic. Most of the bacteria on the food that you're eating is going to be entirely harmless. Even in the case where there may be pathogenic bacteria on the food, it's not necessarily going to cause an infection unless there's enough of them. Remember, I said that most pathogenic bacteria require large numbers of cells to cause a full-blown infection. So that's why things are more dangerous the longer they sit. It can also be caused by other microbes. Viruses can cause food poisoning. Parasites and fungi can cause food poisoning. A lot of the most common foodborne illnesses are caused by bacteria. Well-known examples include E. coli. That's what the E in E. coli stands for. Escherichia. That's the genus name. Another really well-known and thankfully less common than it used to be one is Clostridium botulinum. Anybody know what else we use that one for? Anybody know anyone who's had a Botox injection in their face? Botox is derived from the poison that Clostridium botulinum produces. This is an extremely deadly bacteria that causes very rapid and unpleasant death. And we've managed to isolate some of the compounds from the poison it makes to make this cosmetic product for rich ladies that don't want to have a wrinkle in their forehead. Basically what Botox does, well the poison caused by, or produced by Clostridium botulinum is a paralytic. It causes nerve paralysis or muscle paralysis. So when you inject the derived compound that we now call Botox, what that's doing is it's paralyzing nerves and muscles, preventing things from contracting and making things look much more smooth. So it prevents the wrinkles, but it also prevents you from moving parts of your face. And I think it's really weird that people actually pay money to have that stuff shot in them. Clostridium botulinum is an anaerobic bacteria. What does that mean? Doesn't need oxygen. It also doesn't even survive in the presence of oxygen. So when something is anaerobic, not only does it not need oxygen, but heavy exposure to oxygen will actually kill it. So Clostridium botulinum is commonly found in, or was commonly found in canned goods. Back when we had slightly less effective canning processes, maybe like 100 years ago, this would be a lot more common. However, even these days, if you ever pick up, a can of beans or something, and it's bulging out, do not eat those beans. Because that's generally a sign that something is growing inside that can, and it may be Clostridium botulinum. We're better at the canning process now. We're better at killing the microbes inside the cans when we seal them, so it's much, much less common. That's how canning works, by the way. That's why you don't have to refrigerate stuff in there. Because we seal it, then we apply enough heat to it to kill anything inside of it, and then nothing is left living in there to grow. If it's done properly. Now, in addition to causing foodborne illnesses and food poisoning and unpleasant stuff like that, a lot of prokaryotes are also deliberately used by people to produce a lot of really great foods. Stuff like that. So cheese. Who here likes cheese? If you tell me you don't like cheese, I basically assume you're lying, because cheese is awesome. Cheese is heavily relies on bacterial fermentation in the production process. So the milk has to be acidified by some means in order for it to form that nice, delicious, melty cheese. You can use artificial methods of acidifying the milk, like vinegar or like lemon juice or something, but the best cheeses are produced by bacteria, or produced using bacteria. Salami is another good one. Who here likes salami? Yeah, salami is also great. It's almost as good as cheese. It's really good with cheese. Salami is not cooked. Anybody know that? Yeah, but it's also not raw. I mentioned earlier in the semester that there are more than one way, or there is more than one way to cook something. Heat is what we commonly think of, but you can also cook things with alcohols, you can cook them with acids, you can cook them with bases. In the case of salami, it's cooked, or yeah, I guess cooked is the way to put it. It's cooked by the acidifying effect of bacterial fermentation. So when they're producing salami, there's several ways to do it, but in general it involves hanging the meat up in a hot, humid room and allowing a bunch of bacteria to grow on in the meat, which will then produce acids, which denature the proteins, which make it safe to eat. And then they will hang it in a cool place to dry for a while, and that's why it doesn't need to be refrigerated. Great stuff. Yogurt. Who loves yogurt? Yeah, also produced using bacteria. In fact, these days they're openly advertising in many cases. Anybody use Activia or eat Activia? Yeah, that's promoted as a product that can help promote healthy gut flora. So they didn't used to talk about the fact that there's living bacteria in your yogurt, but now you pay extra for yogurt with fancy bacteria in it. Anyway, yogurt produced using bacteria. Also fish sauce and other fermented seafood products. Anybody here like fish sauce? That's really commonly used in Southeast Asian cuisine. Imparts a really nice umami flavor to the food. That's also, yes? Soy sauce is also fermented. It's different from fish sauce, but it's involved, or soy sauce is produced by basically fermenting soybeans for like 9 months. So that one also involves bacteria. A lot of the foods we enjoy are produced using prokaryotes.
That's ethanol too, though.
Not prokaryotes. What produces ethanol? Anybody here make beer?
Fermentation, sugar.
Using what yeast? Yes. And what are yeast? They are unicellular fungi, which are eukaryotic organisms. So there are plenty of other microbes produced or involved in food production. We use yeast to make beer. We use yeast to make wine, vinegar, bread, all kinds of great stuff. But those are not bacteria. They're not prokaryotes. Now, a little bit more about the relationship between us and prokaryotes. We generally like to think of ourselves as a single organism. But, in a sense, each of us is a complete walking ecosystem. I mentioned there were more prokaryotic cells on you than your own cells. I didn't tell you how many. Roughly 10 to 100 times more prokaryotic cells living on and in you than you have human cells in your entire body. So they're all over your skin, they live in your guts. You also have other microbes that live in your eyelashes. There's this weird parasite that lives in the roots of your eyelashes that basically everyone over the age of 20 has, but it doesn't really cause any problems. In fact, in many cases, we have mutually beneficial relationships with these prokaryotes. Now, when two organisms share a mutually beneficial relationship, meaning it's good for both of them, we have a specific name for that, and that's called commensalism. So we have commensal relationships with many of our prokaryotes. Gut flora is a great example. When people say gut flora, that means the bacteria living in your gut. Now, flora is usually used to describe plants. When people talk about flora and fauna, that's plants and animals. Bacteria were once incorrectly classified as plants, so we still sometimes use the term flora to refer to them. Gut flora are the bacteria living in your gut, generally in your large intestine and small intestine. You have hundreds of different species living in there too. And the gut floral communities will actually vary in what species they contain from one person to another. Certain foods you eat will promote the presence of certain gut flora or harm the presence of others. There have been studies done showing that, for instance, the artificial sweeteners in diet soda actually have a really harmful effect on your gut flora.
Your gut flora, it influences your immune system, right?
Indeed it does, and I'm about to get into that. One of the benefits that these gut flora provide for you, though, is assisting in digestion. They break down molecules that your digestive system alone can't really break down by itself. This also assists in the absorption of several different vitamins, like vitamin K. We rely on our gut flora to help us absorb that as well. They also prevent infection, largely by, in babies and children, they sort of train up your immune system. In adults, they still also aid in immune function as well. So they keep your immune system doing what it's supposed to do, and even beyond that, a well-established colony of healthy gut flora prevents the establishment of potentially pathogenic flora. So if you have all these nice bacteria living on the wall of your intestines, that's going to take up space and not really leave an open niche for potentially pathogenic bacteria to get established. Now, unfortunately, when you take antibiotics to kill pathogenic bacteria, it can also have harmful effects on the good bacteria in your system. So a lot of the time, if people have to take really extreme courses of antibiotics, the doctor will prescribe the kind of informal name for them is poo pills, where they literally give you a pill containing fecal bacteria that you will then swallow, which will help the reestablishment of healthy gut flora communities in your intestines, because you need those. All right, any questions on this? All right, this is the last slide I've got on prokaryotes for this lecture, because we've only got 5 minutes left, so I don't really want to start on fungi. Does anyone have any questions on anything we've done? All right, so Thursday, we'll pick it up again with chapter 13, talking about eukaryotes, and hopefully we'll finish that and get started in the next chapter. No problem. If you didn't collect your exam from me last time we met, I do still have exam 2 with me, so if you pick that up, feel free to come grab it. And if you have any other questions, office hours will be starting right now.